So when I was a kid, I always loved animals. And I had an uncle who was a vet. I thought I might be a vet, but every time we went to his surgery, he was always having to put animals down and stuff, and that was sort of sad, or they were dying when he was doing calf lambing and stuff and things. So I liked animals, so I sort of wanted to work with animals and stuff, so I thought I'd do biology. Never did it at school, but did it at university, and after first year university, I thought, yeah, this is for me. Yeah, I wish I'd learned to do computer programming. And I find I've got some students who are really patient and can go on to, help pages and also email groups and things like this to get help with programming. I never learned programming, and in biology now, programming and bioinformatics is so important, and it's lucky I've got some very good PhD students, so I'd really struggle with that. So to me, I cloned a couple of genes. The whole genome sequenced now, but in the last 10 years, there would be a race to find particular genes, either particular genes associated with diseases, or in my case, DNA binding proteins that regulated those genes. And we'd all be racing to either purify them, sequence the protein and clone them, or to clone them by different means. And I cloned a couple of them, KLF3, and then me and a student cloned one called FOG. And those were fairly big papers at the time because we were in a race to get one particular finish line, and we got there first, and that was pretty exciting. That was sort of like discovering a new species or something.
Okay.
So that's really interesting. This is one of the nicest things about science. I think if I was on a desert island, doing it by myself, I think I'd lose motivation. But when you're working in a team, you're always doing your own stuff, but you have other people helping you with different aspects of it, and that's great. So the lab, the more people you got, the more different things that are happening. Then I've got international collaborations. Every morning when I wake up, I check my phone to see if the results come through, and then I've got collaborators throughout Australia. At the end of the day, I've got one long-term collaborator I've worked at for a long time, who's at Sydney Uni, and we're friends, always worked together ever since 15 years or so. But otherwise, what I'll do is I'll contact the best person in the world, work with them, say, can you help me with this? They usually say yes, work with them, we write up the paper, and we're sort of friends, but I never work with them again. So mostly it's for a reason. We say, let's finish this together, and we do it, and then move on and work with someone else. So collaborations are very dynamic, but it's fun. It's really, because it just doubles how many results you get. It's interesting. Individually, you have to make a decision what you're going to work on, and everyone individually has the initiative, but to bring it to fruition, it'll always be a collaboration. So sometimes you'll be the lead person and other people helping you to get there. Other times someone else is the lead person and you're helping them to get there. But I do think actual scientific creativity is like poetry or songwriting or something. You've got Lennon and McCarthy, but you can tell who wrote which song sometimes. And I think that it's good to have a team to keep you thinking, but individual creativity is the most important thing, and then collaborating to get the job done. The role of Dean of Science, there's a number of responsibilities. There's financial responsibility for the budget, which is really a matter of discipline. We have a certain amount of money and we've got a million things we could do, and eventually I've got to draw the line and say, look, we'd love to do all the other stuff. This is the line. Sorry, folks, we've just got to live within that, otherwise we're finished. And sometimes that's tough because you've got to disappoint people with that. So there's budget responsibility. There's responsibility for vision and direction. I think that a lot of that comes from the bottom up, individual people's ideas, and particularly scientists. But every now and then you have to sort of say, look, I want us to be the best science faculty in the country, and I want us to be recognised for things in the future. So we're going to make a big contribution in the environment, in biomedical research, and those sort of things. So you can sort of focus on that. And then the third aspect of the role is really a ceremonial role, that I have a huge responsibility to represent the faculty, to give talks, to talk to students, to talk to teachers, to talk to politicians, to talk to business people, and to really explain why I love the faculty and think it's so important, and then hopefully other people love it too, and that does happen. So the ceremonial role is one that you don't sort of expect when you're dean. You think that your job will be to do science and to manage science, but actually being a spokesperson like today is a big part of it. So I think molecular biology, because it's sort of a digital discipline, has huge capacity in the future. I think we will get to the stage where we can control the fate of cells, we can turn genes on and off, and I think that will allow us to basically intercede and cure a lot of diseases. We will be able to understand. I don't think we'll ever have full genetic determinism. We won't be able to sequence someone's genome and say, this is what you're going to look like. But in some cases, we'll be able to understand disease risks reasonably well and to give advice on this. So I think we're just beginning with molecular biology. There's a sort of a few areas in science where you don't understand everything because a lot of stuff doesn't make sense. And what I work on, turning genes on and off, we get a lot of results that don't make sense. But I think if we one day can turn all the genes on and off at will, that's the way you'll beat cancer. If you can turn key genes off and beat viruses. People started thinking we should be able to turn HIV virus off. That should be easy. We're still trying. Yeah, so what I do is I always I park up near the lab. I buy a coffee from the coffee cup, and then I go and talk to my postdocs. Got a couple of postdocs in the lab, and I go and sit and drink the coffee and talk to them about things every day. Wander around, just have a look at the PhD students, sing out if they've got any results, or so. They also email me the results. Then, so I usually don't have anything from 9 till 10. Then I'll come down here and there'll be meetings, which are really sort of all updates from the different heads of maths and chemistry and physics and things like that, and the budget and other things, science policy and the university's approach to science. So I'll do that, and then I won't today, but often I'll wander back through the lab when I go to pick up my car in the afternoon before I go home. So, and on Fridays I spend the whole day in the lab. We have lab meeting in the morning. Every student just stands up and says, look, this is what I did this week, this is what I hope to do next week, but this didn't work. Anyone got any ideas? So we do that on Friday morning, and Friday afternoon I just sort of hang around the lab and catch up with people. So look, I had a friend when we were doing honours, and I went on to do a PhD, and this friend didn't, and said to me later, hasn't all the great stuff already been discovered in genetics? And I remember at the time looking and thinking, yeah, it probably has. It's probably all been discovered, because everything I knew had been discovered. And then since that time, and there's an article in the paper said it again, everything's been discovered. But since that time, green fluorescent protein's been discovered, RNAi has been discovered, epigenetics has essentially become clear, oncogenes have been discovered. There's so much, and whenever you look at it, it looks like all the best stuff's been discovered, and it's easy for people to think because you can't see the undiscovered stuff. You can only see the discovered stuff. So the message would be, don't ever think it's all been discovered. Don't even think it for a moment, because it hasn't been.